last week we were looking at resistance to antibiotics or antimicrobials, and one of the things that hopefully by now you're able to appreciate is that the modes of action of drugs or antimicrobials usually will give us an idea of what kind of resistance that we are likely to come across, okay? The point at which that drug works in the microorganism, okay, will give you an idea of how those microorganisms will adapt, okay, so that they are able to resist the activity of the antimicrobial. So for instance, if you look at beta-lactams, how do they work? The activity is tied up in the beta-lactam ring, okay? And how do they get into the cell? They bind onto proteins, okay, in the cell membrane. How do things like glycopeptides work, okay? All that is linked to how the microorganism will be able to reorganize itself, or if mutations were to occur in that area, they would then affect the activity of the microorganisms. So remember, microbial agents, okay, will work by the microorganisms rather will acquire resistance through either inactivation of the drug, okay? They can alter the target. Remember, the drug has to bind to a target so that it can be taken up into the microbial cell. So the microbial cell can actually change the target so that the drug cannot enter. Then we have reduced cellular uptake, okay, whereby if a microorganism was taking in the drug, we have a scenario whereby it is no longer able to take in the drug. Then we have increased efflux. As much as the drug is coming in, the microorganism acquires a way of removing the drug, so that now the effects of the drug on the microorganism are reduced, okay? So this is just a pictorial representation of what we've been saying, okay? You have these drugs will act at different points within the microorganism, okay? So you have like beta-lactams, they will inhibit cell wall synthesis. So expectation is that when resistance occurs, it will occur around working to prevent this inhibition of cell wall synthesis, and we'll be looking at that, okay? We've said it's tied up in the ring. You break the ring or you change the binding sites, okay? Then we have all this. We have things like disruption of the cell membrane, okay? We have inhibition of protein synthesis, okay? We have metabolic inhibition, and all these are actually ways in which microorganisms will work to try and counteract the effect of antimicrobials. We also have inhibition of DNA or RNA synthesis, okay? So let's start, let's look at a few of the common antimicrobials and see how resistance to them occurs. So for instance, this is a common one that we all know. I'm sure you've been learning this right from the time you stepped into School of Pharmacy. You've heard about beta-lactam antibiotics. We know that the activity of these beta-lactams is actually tied up in the beta-lactam ring, okay? So, and it's actually a widely used class. The beta-lactams will act by inhibiting the carboxyl transpeptidase or penicillin binding proteins involved in the late stages of peptidoglycan biosynthesis. Basically, they work to inhibit cell wall synthesis. So resistance to this usually antimicrobial or this antibiotic is common and often will be caused by beta lactamases, which will act to break the beta lactam ring. It can also happen through mutation in the penicillin binding protein, resulting in reduced affinity. So remember, the drug has to get into the microbial cell to affect the cell, okay? But if it is not getting in, then it means that it will not be able to give you the effect, okay? So reduced uptake and efflux are also seen, but they are less significant. So the significant ones are the beta lactamases, which break the beta lactam ring, and then we have the penicillin binding protein, okay? So beta lactamases, as we've said, the activity is tied up in the beta lactam ring. If you do not have the beta lactam ring in this penicillin antibiotics, they will not be effective. And remember, even last week when we were looking at production of penicillins, we said that you have to guard your culture very carefully, because if it gets contaminated with beta lactamase producing microorganisms, you will lose your whole product, okay? So beta lactamases all share the feature of catalyzing the ring opening of the beta lactam moiety. Thus, the structural homology in the terminal diala of the maturing peptidoglycan shared by all beta lactam antibiotics is lost in activating the antibiotic. So the microorganism is actually able to produce this enzyme that will break that ring, okay? Of course, by now you're aware that industry has tried to overcome this problem by coming up with beta lactamase inhibitors such as clavulanate, sulbactam, tazobactam, avibactam, verbobactam, relebactam, okay, and all the bactams, okay? So these are actually usually, they do not have a lot of antibiotic activity, but they help to protect the beta lactam ring, okay? Because as we have said, activity is bound up in the ring, okay? And if you destroy the ring, you will not be able to have activity. So they are co-administered or even co-formulated together in fixed dose combination, and they will prevent destruction of the beta lactam ring, okay? So for instance, you have seen the amoxyl clavulanate, okay? Tazobactam with piperacillin, okay? Sulbactam with ampicillin, all that. Then, so that is one way in which penicillins will acquire resistance, okay? Another way in which penicillin will acquire resistance is through altered penicillin binding protein. Remember we said the penicillin uses the penicillin binding protein to get into the microbial cell, okay? But now the microbial cell has the ability to change these proteins, okay? So that now these microorganisms are not able to take in the penicillin the way they should, okay? And of course, different microorganisms will alter different proteins on their cell membrane. So for instance, Streptococcus, you have penicillin binding protein 1A, 2B, and 2X. When these are altered, you don't have entry of penicillin as much as it should, okay? And many different others will have altered proteins on their cell membrane, okay? So one of the most significant effects that we've had is with methicillin, okay, whereby we have the methicillin resistant Staphylococcus aureus, MRSA, as most of you have heard. By the early 1950s, the acquisition and spread of plasmid-encoded beta-lactamase had blunted the effectiveness of penicillin. So again, one of the issues with the microorganism resistance is that it's transferable, okay? You have these microorganisms transferring resistance from one group to another, okay? Which has become a big issue because now it means that we are getting more resistance to microorganisms. And actually, that is where usually you stress to patient, make sure that you actually finish your dose. Because if you don't, it means that you expose the microorganisms to minute quantities. And if they are able to mutate, it's possible that they can actually acquire resistance to your microorganisms. So what you want is to destroy them completely. You destroy the whole breed, okay, for lack of a better word, the whole colonization of microorganisms so that you don't get any instances of developing resistance, okay? So methicillin, which was introduced in 1959, okay? But by 1960, already there was resistance. So of course, what you have to keep in mind is that these microorganisms are struggling to survive just the way we are, okay? Think of like the time we had the COVID at peak, okay? We got the vaccines were being developed, but immediately after, you start hearing that now uh, they have mutated, okay? We have a different strain, okay? And then, of course, you kept hearing either this strain is more virulent or it's not very virulent and all that. So again, just like us, microorganisms are trying to survive, okay? So if you develop an agent, they are also working to ensure that they can also develop way to ensure their survival. So that's why you have a scenario whereby you develop an agent against the microorganisms, but a year later you actually start getting resistant strains, okay? So again, that is important for you to be able to appreciate. As much as we may congregate and celebrate development of antimicrobial, microorganisms are also working to ensure that they survive, okay? So that's it for penicillins. Next, I want us to look at glycopeptide antibiotics such as vancomycin and teicoplanin, okay? Usually for their activity against the microorganisms, they will bind to the terminal D-alanine side chains of the peptidoglycan and prevent cross-linking in a number of gram-positive organisms, okay? So they are not active against gram-negative organisms due to the presence of the outer membrane. The vancomycin use increased dramatically in response to increasing incidences of methicillin-resistant Staph aureus, and resistance was first reported in the Enterococci in 1988. So again, one of the things I want you to appreciate here is that generally what happens, like in this case, when MRSA was found, and then they realized that vancomycin was a good alternative, everyone decided to go for the vancomycin, okay? And now when you increase the use of an antibiotic out there, you're actually increasing chances of developing resistance. And that's why when we'll be looking later on at some of the policies, okay, antibiotic policies, that is the use of antibiotics. Don't just give antibiotics for anything, okay? Because when you release a drug out there indiscriminately, you're actually increasing the chances of developing resistance, okay? And it can be very distressing when you're in a hospital situation whereby you have a patient, you can see they have an infection, but they don't seem to respond to anything. Okay, you test for sensitivity in the lab and you find that nothing that you actually have I mean, the patient cannot respond to anything that you have. All the antibiotics available, you find that the microorganism responsible for the infection is, you know, resistant to them, okay? So it's a matter of basically watching the patient die under your eye, and there's nothing that you can do about it, okay? So again, this is that is why you want to control the use of antibiotics, okay? So again, with the vancomycin, that is basically what happened. So you'll find that for most of this, like this vancomycin and teicoplanin, you'll find that their use is controlled in hospitals. I mean, it's probably a little too late, but it's good anyway, okay? Because again, we don't want to have excessive resistance throughout the community. Then we have aminoglycoside antibiotics. These are hydrophilic sugars possessing a number of amino and hydroxy substituents. The amine groups are protonated at biological pH. In their polycationic nature, the molecules have a high affinity for nucleic acids, particularly the acceptor A site of 16S ribosomal RNA. So again, remember we said the activity of the antibiotic will give you an idea of how resistance will actually occur, okay? So for instance, this when you look at these aminoglycosides, they will bind to the ribosomal RNA, okay? This interferes with the accurate recognition of the transfer RNA by the ribosomal RNA during translation, and may also perturb translocation of the transfer RNA from the S site to the peptidyl transfer RNA site, P site. So again, you remember from your biochemistry, okay, how the production of manufacture of proteins was happening, okay? You have this RNA, okay, moving, and what happens is if you interfere, okay, with that, then it means that the kind of proteins that you end up producing will not be useful to the microorganism. Remember, every protein that is being produced in your body, okay, is useful. It's being produced for a purpose. So think about something like insulin, for example, okay? Insulin is being produced, and the reason it is being produced is to control the amount of sugar in the blood, okay? Now, if you interfere with the kind of insulin that is being produced, or you just interfere with insulin production, it means that we as humans will not be able to control sugars in our blood, and what will happen, okay? We start having a cascade of issues, okay? So again, I want you to think of it like that in the microorganism. They have proteins that need to be produced, and those proteins serve a purpose. If you interfere with that, you might end up actually killing the microorganism, okay? Just the way if you do not produce the right protein to control sugar in the blood, you could actually end up dying because of that, okay? So high-level resistance in aminoglycoside-producing microorganisms is by methylation of the ribosomal RNA. However, this is not the mechanism of resistance in previously susceptible strains. The most common mechanism for clinical aminoglycoside resistance is their structural modification by enzymes expressed in resistant organisms, which compromises their ability to interact with ribosomal RNA. Okay? So again, structural modifications have been carried out to develop compounds that are not susceptible to modification by these enzymes. And examples are tobramycin and amikacin. Again, as we've said, remember these microorganisms, as much as our aim is to try and destroy them, their aim is try to evade our destructive mechanisms. Okay. So you will find that you develop a product, but the microorganisms, later on you may find that the microorganisms actually are able to develop a way of evading that antibiotic. Next we have tetracycline. Okay. Major mechanism of resistance is efflux and ribosomal protection. The TETx gene encodes an enzyme which modifies and inactivates the tetracycline molecule, although this does not appear to be clinically significant. The TET efflux proteins belong to the major facilitator superfamily. Okay, and these proteins exchange a proton for a tetracycline cation, usually magnesium complex, reducing the intracellular drug concentration and protecting the target ribosomes in the cell. Okay, then we have fluoroquinolone antibiotics. This will bind and inhibit bacterial topoisomerase enzymes. We have DNA gyrase, which is topoisomerase 2, and topoisomerase 4. Okay. And DNA gyrase tends to be the major target in gram-negative bacteria, whereas topoisomerases, both topoisomerases are inhibited in gram-positive bacteria. Okay. So again, each topoisomerase is termed a heterotetramer, being composed of two copies of two different subunits designated A and B. Again, remember from what we said, you look at the mode of action of your antibiotic, it will give you an idea of how, or of the effect of, or rather it will give you an idea of how microorganisms will work to interrupt its activity. Okay. So topoisomerases are located in the cytoplasm, and therefore the fluoroquinolones must cross the cell envelope to reach their target. Okay. So remember, there are those drugs that we've said, like the penicillins, that will work on the membrane. Okay. But there are those drugs that will actually work in the cytoplasm, okay, which means that they have to get in, okay. So again, it's possible that one way of microorganisms ensuring that they resist the effect of these antibiotics is preventing the entry of these antibiotics. Okay, so for instance, with the fluoroquinolone, since they have to get to the cytoplasm to inhibit the topoisomerases, usually what will happen is you can actually get a scenario whereby they are permeability to this fluoroquinolone is reduced. Okay. Efflux makes a contribution to resistance. Okay. Then we have macrolides, lincosamide, and streptogramine. These are what you've had referred to as MLS antibiotics. They inhibit protein synthesis by binding to a target site on the ribosome, even though they all differ in structure. Okay, gram-negative bacteria are intrinsically resistant due to their permeability barrier of the outer membrane. Three resistant mechanisms have been described in gram-positive bacteria. We have target modification, okay, then we have efflux, then we have ribosomal mutation. Okay, so again, remember what we said. Mode of action will have a bearing on the resistance, okay? Because like in this case, we are talking about modification of the target, whereby involving adenine methylation of domain 5 of the 23S ribosomal RNA. And ideally, this are supposed to bind here, but now when you modify it, they can't bind, okay? They are not able to work unless they are inside the microbial cell. What does the microbial cell do? Efflux. It removes the drug. As it's moving in, it's being removed, okay? Then ribosomal mutation, okay? Again, remember we have said that this they interfere with protein synthesis, okay? And if you get mutation in the ribosomes, this will not be able to bind and interfere with that synthesis. Then we have chloramphenicol. This inhibits protein synthesis by binding onto the 50S ribosomal subunit and preventing peptidyl transferase step. Decreased outer membrane permeability and active efflux have been identified in gram-negative bacteria. However, the most major resistance mechanism is drug inactivation by chloramphenicol acetyl transferase, okay, which occurs, of course, in both gram-positive and gram-negative species. Then we have oxazolidinone antibiotics such as linezolid. It has a novel target in protein synthesis. Linezolid does not interfere with translation in initiation at the stage of mRNA binding or formation of 30S pre-initiation complexes. Rather, it involves binding the 50S ribosomal RNA. Its affinity for the 50S RNA, ribosomal RNA, from gram-positive bacteria is twice that for the corresponding molecule in gram-negative bacteria, and as such, linezolid has been approved for treating various gram-positive infections, including MRSA. Resistance is appearing, although rare at present, and it's due to mutation in the central loop, domain 5 of the component 23S ribosomal RNA subunit. So again, when you get into practice, these are some of those molecules that you'll find are not, they are sort of controlled, okay? Because one of the things that you'll realize when you get into practice is that sometimes people just want to prescribe. And you may find that this is even encouraged by companies, the companies producing these drugs. Of course, you know, the reason they are doing that is so that they can get sales, okay? But the truth is, if you do not control some of these drugs, you may get a scenario whereby you have a patient who is coming in with a resistant bug and you have nothing to treat. So you'll find that in practice, you may have some of these drugs that have been placed in sort of controlled, okay? Like I remember one of the places I worked, we had some of this controlled, and we would say that you had to, if you had to prove that your patient could not benefit from any other product, okay? Of course, again, apart from the fact that we wanted to limit the use, we also did that because the cost was very high. Because you come and find a patient can benefit from a drug that is going to cost them 500 shillings, and yet you get a clinician prescribing a drug that is going to cost them 5,000, okay, which doesn't make a lot of sense. Then we have trimethoprim, which acts by competitively inhibiting dihydrofolate reductase. Resistance to this trimethoprim can be caused by overproduction of a host dihydrofolate reductase, mutation in the structural gene for dihydrofolate reductase, or acquisition of the DFR gene on coding a resistant form. Okay, so again, as we said, look at the mode of action. It will give you an idea of how resistance to this antibiotic or antimicrobial will be acquired. Okay. Acquisition of DFR gene encoding for alternative dihydrofolate reductase of type 1, 2, or 4 or 5 is the most common mechanism of trimethoprim resistance among the Enterobacteriaceae. Then we have peptide antibiotics such as polymyxin. They include the polymyxins, bacitracins, gramicidins, as well as the glycopeptides. Polymyxins and other cationic antimicrobial peptides have a self-promoted uptake across the cell envelope and perturb the cytoplasmic membrane barrier. Addition of a 4-amino deoxyl L-arabinose moiety to the phosphate groups on the lipid A component of gram-negative lipopolysaccharide has been implicated in resistance to polymyxins. So generally, that is resistance to some of the common antibiotics that you will find in practice. Okay. Another aspect of resistance that I want us to look at is the resistance to the antimicrobacterial therapy. Okay. So what we commonly refer to as TB. Okay. We, of course, by now you know that we are concerned with TB sensitivity to TB medications because of the impact, the community health impact or the public health impact. Okay. So microorganisms in TB infections tend to grow slowly in a near dormant state with little metabolic activity. This nature of mycobacterial infections means that chemotherapy differs from other infections. A number of conventional antimicrobial targets are not suitable. And by now, hopefully, you're aware that we treat TB using several drugs, okay? Because of this near dormant state, they are not very sensitive, okay? So you have to ensure that you use combination therapy just so that you're able to ensure that you can actually target these microorganisms and reduce their numbers, okay? And get to control them, okay? So currently the first line, the main first line combination, we have isoniazid, which is bactericidal. Its aim is to reduce the count of aerobically growing organisms. Then we have parazinamide, which is active only at low pH and makes it well suited to killing organisms within necrotic foci early in infection, but less useful later on when these foci have reduced in number. Okay, so again, this gives you an idea of why usually pyrazinamide will be used during the first phase of TB treatment, but later on it is not used, okay? Because remember, we are saying it is effective at low pH, okay? But when the necrotic foci have reduced, then it is no longer effective, okay? Then we have rifampicin, which targets slow-growing organisms, okay? Then we have ethambutol. Current treatment regimens have a high cure rate, and the combinations of agents makes it highly unlikely that there will be a spontaneous resistant isolate to all the components. Problems most commonly occur in patients who receive inadequate therapy, which provides a serious selection advantage. So remember, again, we said that one of the reasons why you encourage patients to finish the antibiotic dose is so that you don't get a scenario whereby you get microorganisms that are introduced to an inadequate dose, so they get a chance to actually mutate and acquire resistance to your antimicrobial. Okay, so it's the same case here with mycobacterial therapy. You want to ensure that the patients are actually getting the full therapy to ensure total eradication of the microorganisms from the system, so that you don't get any microorganisms that were exposed to some therapeutic levels and therefore given a chance to actually mutate and multiply. Okay. Resistance mechanisms have now been described, and multiple resistance poses a serious threat to health. Resistance can occur to single agents and subsequently to multiple agents. So let's look at resistance to this antimicrobacterial therapy. Resistance to rifampicin has been documented, and it arises from mutation in the beta subunit of RNA polymerase encoded by RPOB, and resistant isolates show decreased growth rates. Modification of the catalase gene results in resistance to isoniazid, mainly by reduced or absent catalase activity, which is required to convert isoniazid to the active hydrazine derivative. So again, from your PharmChem, hopefully it's been mentioned that isoniazid is actually a prodrug, okay? And when it gets into the body, it needs to be activated, okay, through catalase activity, okay, to the hydrazine derivative. But now, if the microbiome is actually modifying the catalase gene, it means that the catalase produced is not able to actually activate isoniazid, okay? Then we have low-level rifampicin resistance, which can be obtained by point mutations in INH, leading to its overexpression. Okay. Then we have pyrazinamide, which is also a prodrug. Okay. It requires pyrazinamidase to produce the active pyrazinoic acid. Okay. So when you get a mutation in the pyrazinamidase gene, again, you will interfere with this process of activation. Then we have streptomycin, which can arise from mutations in RRS and RPSL, which affect streptomycin binding. This accounts for half of the resistant isolates. Okay. So what this tells you is that, again, remember these resistant mechanisms are basically things that have been studied, okay? But you may find that, like in the case of streptomycin, this has been documented, but what they're saying is that it does not account for the full resistance, okay? So there are other resistance mechanisms that have not been documented. Okay. Then we have ethambutol. Resistance has been noted in Mycobacteria tuberculosis and other species such as Mycobacteria smegmatis. Ethambutol inhibits polymerization of arabinine. Ethambutol inhibits the polymerization of arabinine in the arabinogalactan and lipoarabinomannan, okay, of the mycobacterial cell wall, okay. Then we have missense mutations in the MB gene. In this locus will confer resistance to ethambutol. So we've looked at individual resistance to these antimycobacterial drugs. Next, I want us to look at multiple drug resistance, because this has been an issue, especially with tuberculosis, whereby you have, as much as we've said, use of multiple drugs is very helpful to prevent resistance and to encourage to treat the condition. What has been realized is that there is actually multi-drug resistance, whereby you get resistance not just to one drug, but to multiple drugs, okay? And this can actually be an issue. So this multiple drug resistance is propagated through the following. We have R factors, mobile gene cassettes and integrons, chromosomal multiple antibiotic resistance, MAR locus, and multi-drug efflux pumps, okay? So we'll look briefly at this, but we won't go into the nitty-gritties of it. So MRSA can harbor both small, for us to be able to appreciate how this multiple drug resistance occurs, let us look at MRSA, okay? And that will help us to be able to appreciate that. So MRSA can harbor both small cryptic plasmids and larger plasmids, encoding resistance to antiseptics, disinfectants, and all those antibiotics and antimicrobials, okay, including multi-drug resistant Mycobacteria tuberculosis, okay? Instances where isolates can become resistant to multiple chemically distinct agents in a single biological event are of great concern. One of the earliest examples was in Japan in 1959, where previously sensitive E. coli became resistant to multiple antibiotics by acquisition of a conjugative plasmid. So again, going back to your if you remember from your you did this unit in with Dr. Julia, okay? This is where you have genes, okay? Microorganisms are able to actually transfer genes from one biotechnology is what I'm talking about, okay? You're able to transfer genes from one microorganism to another, okay? Now, that with the biotechnology, that can be done scientifically, okay, by us manipulating the microorganism. But of course, hopefully you are able to see in biotech that microorganisms have the ability to actually pick some of these genes themselves, okay? And in this case, like the one we are talking about here in Japan in 1959, E. coli, okay, acquired resistance through acquisition of a conjugative plasmid from resistant Salmonella and Shigella isolates, okay? A number of R factors have now been characterized, including RP4, R1, and PH6, okay? And all these will encode for different resistance to different antibiotics or antimicrobials. Okay, so next I want us to look at clinical resistance. Uh, what does it mean when you have resistance clinically? How do you decide that this is clinical resistance? Okay, and at what point do you consider it significant? Okay, so MIC value is the lowest concentration of an antibiotic at which bacterial growth is completely inhibited. Okay, this is usually determined in the lab. The minimum inhibitory concentration value. Okay, it's determined in vitro in the lab. Then we have breakpoint value. This is the concentration at which bacteria are susceptible to successful treatment with an antibiotic. Okay. This is usually determined in vivo, okay, within the human body. So MIC in the lab, okay, the lowest concentration of an antibiotic at which a bacterial growth is completely inhibited. Then we have breakpoint value, the concentration at which bacteria are susceptible to successful treatment with an antibiotic. This is in vivo, okay. So the breakpoint value can be difficult to determine. Resistance mechanisms typically lead to an increase in MIC values. The increase in MIC values does not always equate with clinical failure. And if the MIC value remains below the breakpoint value, then the antibiotic will remain effective in the clinic. So remember, we've said with the MIC, it's determined in the lab, the lowest concentration at which the bacteria can grow. Then we have breakpoint value, which is in the body, okay? And it's the concentration at which bacteria are susceptible. So if you have MIC being higher than your breakpoint value, then it means that in the clinic, this antibiotic is still effective, okay? MIC values are typically determined when the isolate is growing in complex sensitivity test broth. This may not fully equate with sensitivity of the organism when growing in the many subtly different environments encountered during infection in vivo. So again, think about infection. When you're growing microorganisms, you're basically giving them growth media, okay, in the lab. But in the body, think of microorganisms in different areas within the body. Some will prefer to go into the lungs, for example, because they thrive probably in excessive oxygen. Okay, we have those that will go into the blood. Okay, we have those that will go into the bone. We have those that will go into different areas within the body. So again, they go into an area where they are able to colonize and grow successfully. Okay, so that's why we are saying that their growing in the body may not always reflect what you're growing in the lab. And that's why it is very hard for us to equate MIC with sensitivity in the body. That's why we are looking at breakpoint value. And as it is right now, if your MIC is if your MIC value, okay, remains below the breakpoint value, then the antibiotic will remain effective, okay? But if it goes above the breakpoint value, it means that your antibiotic will no longer be effective, okay? So there have been cases of treatment failures despite apparent sensitivity in the test tube, and this resistance is referred to as phenotypic, okay? Because phenotypic resistance is a consequence of adaptation of the organism to grow and survive within the in vivo environment, and subculture into conventional laboratory growth media rarely shows existence of resistant mutants. So one of the things that you appreciate is that when microorganisms show resistance, okay, when they are no longer responsive to an antibiotic, usually what happens is either they have acquired resistance remember, like the mechanisms that we've talked about, okay? Maybe they have altered the target, okay? Maybe they are effluxing the drug. Maybe they are actually, acquired characteristics that are destroying the drugs, all that, okay? Either that or it's phenotypic, whereby when you subculture it in the lab, okay, it doesn't show resistance. Okay, in the body, it's not responding. But when you isolate it and grow it in the lab, it is responding. Okay, that is what we refer to as phenotypic resistance. There are no resistance genes, okay, or resistant mutants, but it's just that the microorganism is able to sort of hide or use the body to evade the antibiotic. Okay, there are several key factors at play here, particularly slow or no growth, nutrient depletion, and mode of growth. Okay, there is a tendency for nutrient depletion, such as restricted or non-availability of an important nutrient, slow or no growth to be associated with reduced susceptibility to antibiotics and biocides. So remember, uh, for some of these mechanisms, remember we saw that you can have an antibiotic that is affecting metabolism of a, a microbial, okay? But if that microbial is dormant, okay, it is dormant, it is not metabolizing at the rate that it should, it means that even if you administer an antibiotic, it will not affect this microbial, okay? It will not affect this microorganism. So in the body, you may have it as not responding to the antibiotic, simply because the normal functioning of the antimicrobial have either been halted or slowed down. Okay, so that if the antibiotic is going to interfere with cell division and yet there is no growth that is happening, then it means that you will see no effect. Okay. Um, Another aspect I want us to look at is what we refer to as bacterial biofilms. Okay. Um, Sorry. Bacterial biofilm. This is a population of cells growing as a consortium on a surface and enclosed in a complex exopolymer matrix. These biofilms can result in medical device-related infections, such as those associated with artificial joints, prosthetic heart valves, and catheters. Some chronic infections not related to medical devices are also due to bacteria either not growing and relatively dormant or growing slowly as biomasses or adherent films on mucosal surfaces. Growth as biofilm almost always leads to a large increase in resistance to antimicrobial agents compared with cultures grown in conventional liquid media, but there is no generally agreed mechanism to account for this resistance. So again, you can imagine in this case, these microbial cells are growing, okay, and then they are enclosed within an exopolymer matrix. So that means that this antimicrobial is not able to penetrate into those matrix, into that exopolymer matrix. Therefore, the microorganisms are protected. Okay, so that even if you give the antibiotic, since it is not able to penetrate that exopolymer matrix, the microorganisms are actually protected. Okay, and as we've said, these biofilms usually will happen around medical devices, okay, such as catheters, artificial joints, prosthetic heart valves. The general resistance of biofilms is clearly phenotypic. Their antibiotic susceptibility is also affected by their growth rate or specific nutrient limitation. The resistance mechanisms may include lack of antibiotic penetration, inactivation, efflux, and repair. There is, however, no compelling evidence that they are uniquely responsible for biofilm resistance. A reduced growth rate probably has an involvement, particularly in that it is associated with responses to stress. During stress responses, key structures are protected and cellular processes close down to a state of dormancy, and it has been proposed that the exceptional vegetative cell dormancy is the basic explanation of biofilm resistance. So again, just for you to note that resistance can be due to biofilms, okay? There is no specific explanation or definite explanation of how this occurs, but these are basically just postulations of how it may occur. But it's important for you at least to be aware that if a patient has prosthetic medical devices, then these are things that you need to be aware of. Okay. So that brings us to the end of resistance to antimicrobials. Next, I want us to look at antibiotic policies and disinfection policies. When we talk about antibiotic policies, remember, as we've kept on saying, when the first penicillin was discovered, it was a big thing, okay? Because remember, initially people would just die because of infections. If you got an infection, you basically just, it would eat you up and you would die, okay? People getting tuberculosis, they would basically just, waste away and die. But then when antibiotics came, it became a life saver, okay? But unfortunately, immediately antibiotics were discovered, as we have said, microorganisms were also not dormant. They've also been looking for ways to survive. Okay, so they keep coming up with ways that are counter effective or counteracting the effect of these antibiotics. Okay, and the misuse of antibiotics by humans has not helped. Okay, because now what has happened is, as much as antibiotics are out here, they're life-saving, people are using them for everything, even for what does not need antibiotic. And what happens? Now you expose these microorganisms to your defense, and what happens? They are able to acquire resistance to this defense. So that is what we are talking about when we are looking at antibiotic policies. What policies have been put in place to ensure that at least the few antimicrobials that we have in practice can last us, okay, a lifetime, even as we look to produce others, okay? So this is a case. If we look at this, this is a case that happened in 2017. Okay, about a 70-year-old American woman who had gone out of the country, and then they came back. They had acquired an infection. Okay, and in this case, they are referring to it as a superbug. Okay, this is a 70-year-old from Nevada returned to the US in August with an infected swelling in her right hip after a long stay in India. This is 2017. Okay, now what they realized is that the patient eventually the patient died, okay? Because unfortunately, this superbug was not responding to anything. All the analysis found the superbug was resistant to all 26 available antibiotics in the US, including the drug of last resort, colistin. Okay, so this is this scenario that I was trying to describe earlier, whereby you have a patient in your care and there is basically nothing you can do about them. Okay, they are dying right under your nose, and none of the drugs that you have can help them. Now, one of the things that you have to guard against, of course, is you can imagine what would happen if this infection were to be transferred to other patients. Okay, it's the same case as the multi-drug resistant tuberculosis. Okay, sometimes you know the law says that you have a right to choose whether you want to be treated or not. But if your refusal to be treated is a risk to other Kenyans around you, okay, then is it still just your individual choice? Because you can imagine it costs, right now, I believe it costs a number of uh, thousands to treat tuberculosis, okay? And if you look at multi-drug resistant TB, it costs about 2 million, okay, to treat one patient for the full duration, okay? So you can imagine if this one patient were to be released into the community and they decided, I'm not going to take drugs, and then that infection spread to the rest of the community, you can imagine the effect, okay? That's what you want to guard against. Because think of this uh, patient, okay, who later died. They had a superbug that was responding to nothing. Imagine if this superbug were to spread, okay? This would be quite sad, okay? So with that in mind, antibiotic policy consists of prescribing strategies to optimize indication, selection, dosing, route of administration, duration, timing of antibiotic therapy to maximize clinical cure or prevention of infection whilst limiting the unintended consequence of antibiotic use, including toxicity and selection of resistance microorganisms. The goal of antibiotic use policies is to reduce antibiotic usage and encourage the return of susceptible human commensal flora. So what are some of the consequences of antibiotic misuse? First, we have antibiotic resistance, which we have talked about, okay? You can have increased infection severity, like the case of the lady that we have talked about, okay? We have increased cost of treatment because remember, usually you have the first line of treatment, which generally is an affordable treatment, okay? But as you get into more resistant strains, you start getting into controlled products, okay? Whereby the cost may actually be very high, okay? So you can find that your normal cost of, say, amoxicillin out in the chemist might be 500 shillings, okay? But if you get resistance to such and you decide to now go for a product like assuming your patient now is only using linezolid, okay? Or you get to a point where maybe you decide your patient is only responding to meropenem. If you look at the price of meropenem right now, it's usually meropenem is given 3 times a day, okay? The price of meropenem per vial is actually around 5,000 shillings. Okay, so 5,000 shillings 3 times a day, that is 15,000 shillings per day. Okay, assuming you're going to put this patient on this drug for 10 days, you can imagine from the 500k of amoxicillin dose for the full course, now you're talking about 150,000. Okay, so that's increased cost of treatment. Then we have side effects. Again, one of the things that you'll appreciate is that when you're choosing drugs for first line, generally one of the things that you will look at, and hopefully you're able to remember this, we did this in fourth year when we were looking at dispensing, okay, and formularies, and we were looking at what informs the choice of a drug into a formulary. Okay, one of the factors that would be considered is side effects. Okay, so that generally drugs that are used first line usually will have fewer side effects. Okay, but when you get into things like super infection, okay, uh, you here you're looking at a scenario whereby your aim is basically just to save the patient. Okay, you're not thinking of the side effects. You're thinking, let's save the patient because we have no alternative. Remember, like last week when we were talking about the scenario of the baby who came in and the infection they had was responding to nothing except ciprofloxacin. What do? Do you watch the patient die? Okay, of course, we did not do that. We decided to give the baby ciprofloxacin, then worry later about what else can happen. Okay, so those are side effects we are talking about. With your first line, you're not really concerned so much about side effects. For this baby, there are other available options, okay, that were available in pediatric formulation. Problem is they were not effective. The infection the baby had was not responding to any of them. What do? We take the next available effective product, okay, even though we know that it may not be the recommended product. Okay, so then we have rehospitalization. Patients administered with inappropriate antibiotics are at increased risk of rehospitalization. Okay. Then we have, okay, moving on with antibiotic policies. The more antimicrobial agents presents both an increasing problem of selection to the prescriber and difficulty for the diagnostic lab as to which agents should be tested for susceptibility. Some classes, such as penicillins and cephalosporins, have a large number of drugs, which can be overwhelming in terms of which ones to choose to test for susceptibility. One of the things that you're going to appreciate when you get into practice is if you say if you went to a private facility, because we know generally government the products that you get may actually be controlled by KEMSA. But if you get into a private facility where you have to make a choice of what needs to be stocked in the pharmacy, okay, think of even let's look at a simple scenario like analgesics. Okay, how many analgesics do you know? Okay, think of paracetamol. Come to the NSAIDs like clofenac, aceclofenac, ibuprofen, all those. There's a whole class of them. Okay, the question is, do you keep all of them? Okay, when we talk about penicillins, are we going to keep all the penicillins in the pharmacy? Okay, are you going to keep all the cephalosporins? think of cephalosporins in terms of first generation, second generation, third generation, now up to fifth generation. Are you going to keep all those? And yet among those, you have several in each generation, okay? So that presents a challenge, okay? An antibiotic policy gives guidance to the prescriber on the choice of agent from a range of agents appropriate to his or her needs and should be supported by laboratory evidence of susceptibility to these agents. Increased awareness of the cost of medical care has led to a major review of various aspects of health costs. Pharmacy budget has often attracted attention as unlike many other hospital expenses, it's readily identifiable in terms of cost and prescriber. Thus, an antibiotic policy is also seen as a means whereby economic burden of drug prescribing can be reduced or contained. So one of the things you'll realize, if you're not careful in the pharmacy, everyone will come and tell you, oh, I want you to keep cephalexin, I want you to keep cefadroxil, I want you to keep. So everyone comes with their own preference, okay? And then you realize that each of these 10 doctors all want a different drug, okay? And remember, you have a specific budget. You cannot be able to purchase all this at once, okay? So sometimes these antibiotic policies will be helpful in controlling such scenarios, okay? Whereby now you can decide, we have this budget, okay? We are going to decide that we will keep maybe one drug in each generation of cephalosporins, okay? How do we decide on that one drug? We will look at sensitivity, okay? We will look at cost, we will look at availability and all that, okay? So these policies will be helpful in that. The cheaper of two compounds should be selected where agents are similar in terms of efficacy and adverse reactions. Generic substitution is also desirable, provided that there is bioequivalence. It's impractical for pharmacists to stock all the formulations of every antibiotic currently available. Therefore, an antibiotic policy can produce significant savings by limiting the amount of stock that is held. A policy based on a restricted number of agents also enables price reduction on purchasing costs through competitive tendering. So for instance, think of a scenario whereby I have a pharmacy, I'm buying cephalexin, 50 tablets, cefadroxil, 50 tablets, and then think of a scenario whereby I decide, okay, I'm just going to buy cefadroxil, and instead of buying 50-50, I'll now buy 100, okay? You can see with 100, it's possible for me to negotiate for price, okay? Such that the bulk, if I get bulk, I can actually carry out competitive tendering and get reduced prices. It's important to note that the occurrence of drug-resistant bacteria within an institution can cause a big problem. The presence of sick patients and the opportunity for the spread of resistant microorganisms can produce outbreaks of hospital infection. Excessive use of selective agents has been associated with emergence of drug-resistant bacteria, which have often caused serious problems within high dependency areas such as ICUs or burns units where antibiotic use is often high. Okay, so again, these policies will help you to be able to prevent such scenarios, okay? And usually some of these policies are very useful, especially when you're talking about high dependency areas. ICU, what is your antibiotic policy for ICU, okay? Are you just going to use one agent throughout, okay, for the whole year, Okay, what is the implication? How about your burns unit? What is your what is your policy for antibiotics? Uh, uh, Probably a number of you are able to hear about this story, okay, very sad story that happened in Kenyatta National Hospital in the newborn unit, okay, whereby 11 newborns actually died in that newborn unit, and the suspected to be Klebsiella. Okay, the babies were actually dying because of cross infection, cross contamination. This infection was moving from one baby to the next to the next. Okay, and 11 newborns actually died before somebody raised the alarm. Okay, this was captured in 2019. Uh, so you can imagine it's quite unfortunate that such a thing can actually happen. But if you don't have antibiotic policies, you don't have infection control policies such that a nurse or a doctor should not move from one baby to the next without washing their hands, without disinfecting. Because how do you explain that Klebsiella killed all the 11 babies in the newborn unit? Okay. I mean, it's quite sad. It means that this is a microorganism that was actually doing rounds in the newborn unit. Okay. So there has been a reported case of a multiple antibiotic resistant Klebsiella within a neurosurgical intensive care unit in which the organism became resistant to all currently available antibiotics and was associated with the widespread use of ampicillin. On restriction on the use of all antibiotics and in particular ampicillin, the resistant organism rapidly disappeared and the problem was resolved. So you can see why we need antibiotic policies within this high dependency unit, because you can imagine if every patient who is coming in is just being given ampicillin, and then now in this case Klebsiella aerogenes was found to be in this unit and it was resistant to ampicillin. Okay, of course, when these guys at least realized, they decided to change, limit the use of antibiotics in that unit, and especially with ampicillin. Okay. Currently, the most important hospital-acquired pathogen is methicillin-resistant Staph aureus, which is responsible for a range of serious infections such as pneumonia, postoperative wound infection, and skin infections, which may in turn be complicated by bloodstream spread. The use of vancomycin and teicoplanin has escalated as a consequence and in turn has been linked to the emergence of vancomycin-resistant enterococci. So again, remember we said one of the reasons why you limit the use of an antibiotic is to prevent such an occurrence, okay? Because when MRSA came up, people decided, oh, now let's use vancomycin and teicoplanin. The problem is, with the degree of use that was being used for methicillin, it's the same degree that they went into vancomycin, okay? And now because of that, we have vancomycin resistant enterococci. So again, as I mentioned, for most of these hospitals, you will find that this is a controlled molecule, okay? You don't just use it because you don't want a scenario whereby you get resistance to these molecules or widespread resistance, okay? In formulating an antibiotic policy, it's important that the susceptibility of microorganisms be monitored and reviewed at regular intervals. This applies not only to the hospital as a whole, but to specific high dependency units in particular. Remember, we've said high dependency units, it's very important that you actually do this monitoring. Okay, we are talking about newborn units, we are talking about burns unit, we are talking about high ICUs, we are talking about HDUs, all these places where you have high dependency, and in more often than not, you will find there's a heavy use of antibiotics in these areas. Okay. This will provide accurate information on drug susceptibility to guide the prescriber as to the most effective agent. It will also curb the spread of resistant microorganisms in an institution. So, what kind of antibiotic policies are we talking about? There are different approaches to formulation of antibiotic policies. They can range from deliberate absence of any restriction, where you basically say everyone can prescribe the way they want. Okay, you assume that every clinician went to school, they were taught about antibiotics, they are free to choose what they can prescribe. Okay, if they choose to prescribe vancomycin as first line, well and good. If they choose to prescribe linezolid or meropenem as first line, that's up to them. Okay, and so this can range from deliberate absence of any restriction on prescribing to a strict policy whereby all anti-infective agents must have expert approval before they are administered. Okay. So, like, I remember a scenario whereby one of the places I worked, remember I told you that the cost of meropenem at that time, I've not updated to see how much it is right now. It was costing 5,000, okay, per vial. And assuming you're giving the patient 3 times a day, that's 15K per day, okay? And some of these patients will be on it for 10, 14 days, okay? So you can imagine this is a patient that you're going to charge 150K. Now, one time we had a visiting intern from the US, and they came in, and they basically first day on the ward, they got a patient, they prescribed meropenem. Okay, so but uh, they had not been briefed on our policies. Okay, antibiotic policies. So when we called this prescriber in and explained to them, because one of the things I think where she was coming from, uh, this would just be paid. It was automatic, either the government or insurance would cater for this. Okay, the patient would get the drug, there would be no restriction. But in the scenario where I was, this is we need you to pay this upfront. Okay, and when we asked her whether they had done any sensitivity tests, basically nothing had been done. She just looked at diagnosis and decided, I'm going to treat with meropenem. Okay, the patient ended up being treated with a simple antibiotic that was going to cost them less than 1,000 shillings. So you can see the importance of having antibiotic policies. Okay, they can prevent scenarios such as this, because you can imagine if you're the patient, and it's not that there's no other drug that can treat you. There are available medicines that can treat you at much lower cost, and yet a clinician is giving you a bill of almost 200,000 Kenyan shillings. Okay, and yet you could have paid just 1,000. Okay, so that's what we are talking about. So there are different policies. We have restrictive policies, which can be of two main categories, according to whether they are mainly laboratory controlled by employing restrictive reporting, or they are mainly pharmacy controlled by restrictive dispensing. Okay, in many institutions, it's common practice to combine aspects of the two approaches. So based on those restrictive dispensing and restrictive reporting, there are four main types of antibiotic policies. We have free prescribing, restricted reporting, restricted dispensing, and combined restricted reporting and restricted prescribing. Of course, we said that this is usually the ideal, combined restricted reporting and restricted prescribing. So let's look at each of these and see what they imply. Free prescribing. The advocates of a free prescribing policy argue that strict antibiotic policies are both impractical and limit clinical freedom to prescribe. So there are people who believe that they have gone to school, they should be free to make their own decisions, and they can basically prescribe whatever they want. It's also argued that the greater the number of agents in use, the less likely it is that a drug resistance will emerge to any one agent or class of agents. Okay. So like the case where we've talked about with the ICU, okay, whereby we had everyone prescribing methicillin, ampicillin until now when they got resistance, okay. They realized that they had been using ampicillin for so long, they got a resistant strain that was common within that ICU, okay? They decided to limit the use of ampicillin and then also limit use of control the use of antibiotics. And when they did that, they were able to eradicate that uh, resistance strain to ampicillin, okay? This is the argument that they are using here, that if you just have a few agents that you're using, it's very easy to get resistance to them. But if you release all the agents out there at once, then it's unlikely that you will develop resistance against one agent or a class of agents, okay? So that is their argument. This free prescribing policy, however, is not universally supported since it could spell disaster. You can imagine if we were to get resistance to all these agents at once, okay, we would be in trouble, okay? So you don't want a scenario like that. The number of available antibiotics is limited, and applying no checks or controls could result in cases of superbugs unresponsive to any available drugs. So you don't want a scenario where, because this argument where you want to release all agents into practice is assuming that we have so many antibiotics in practice that this could not happen, okay? But the truth is the antibiotics available are not many, okay? And should we have a scenario whereby we get resistance to all of them, it would spell disaster, okay? So that is free prescribing, not your ideal policy. It's not supported. Then we have restricted reporting, whereby laboratory usually will test a limited range of agents against bacterial isolates. So the lab will decide, we are going to say, take test against penicillin, cephalosporin, first generation, second generation, third up to fifth. We pick one agent in each generation, test against it, okay? And then pick one and recommend that this is the one that is microorganisms are sensitive to. Therefore, these are the ones that you will stock in the lab. They can go about it like that, or they can decide drugs that are used to treat different sites, okay, in the body. So for instance, you have drugs that are generally accepted for UTIs, drugs that are generally affected for upper respiratory tract infections. We look at them and then we pick a number, test against them, and then now pick a drug and decide this is what we are going to stock, okay? So the practice of testing a single agent to represent a specific generation such as first, second, third generation compounds is questionable. And with the new compound, susceptibility should be tested specifically to that agent. By selecting a limited range of compounds for use, sensitivity testing becomes a practical consideration and allows for the clinician to use such agents with greater confidence. So at least if you tell the clinician that based on the kinds of infections we've been getting, we have tested them against, these are community-acquired infections. They have been tested against, say, trimethoprim and have been found to be sensitive to this. Clinicians are able to use them with confidence, okay? So that is restricted reporting. Then we have restricted dispensing. Involves restrictions on dispensing on antibiotics pending expert approval, okay? So someone comes to the lab, the pharmacy, to get a drug dispensed to them, but you tell them, no, you have prescribed linezolid, we are not going to give it to you. You wait for expert approval. Okay. In this case, usually with these institutions, you will find that these are large institutions where you have a microbiologist or an infectious disease specialist who is allowed to give approval for antibiotics. Okay. So if you come in with a prescription, we have to call the expert for approval. If they don't approve, then we don't dispense. Okay. In such a case, the expert opinion may be provided by the microbiologist or infectious disease specialist. Such a system can only be effective in large institutions where staff are available 24 hours a day. So you can imagine if you only have one expert to give approval and sometimes they are not there, okay, that would be a big issue. But if it is a big institution and we have several experts and they can be reached at any time of day or night, then that could be possible, okay? However, this approach is often cumbersome, generates hostility, because you can imagine one clinician prescribed and then someone else has to go and approve, okay? It could actually generate hostility, okay? It does not necessarily create the best educational forum for learning effective antibiotic prescribing. This policy can be modified to an approach which involves dividing agents into those approved for restricted and those for unrestricted use. So like this is a version that we had in the institution I was telling you about, okay, whereby we had those antibiotics that were for first line everyday use. Any of the clinicians were allowed to actually prescribe them. They were affordable, okay? And then we had the restricted ones that were some of the broader spectrum. They were quite expensive and they were controlled, okay? So for those, in our case, we had those that if you write a prescription, no one has to approve it. And then we had those that if you wrote a prescription, it has to go through the lab. You have to show us sensitivity tests to show that you're getting this antibiotic because the infective microorganisms are not responding to any other antibiotic and they are responding to this antibiotic that you have recommended, okay? So like in our case, ours was combined reporting and combined restricted reporting and restricted prescribing, because what we were doing is we had divided the antibiotics into groups, and then we required for you to get approval from the lab by showing us sensitivity, okay? So agents are in combined, agents are divided into restricted list and unrestricted list. In this sense, the unrestricted list okay, are appropriate for majority of common clinical situations. This is everyday prescribing. The restricted list may include agents where microbiological sensitivity information is essential, such as for vancomycin and certain aminoglycosides, and carbapenems like meropenem. In addition, agents that are used infrequently but for specific indications, such as parenteral amphotericin B, are also restricted in use. Other compounds that may be expensive and used for specific indications, such as broad-spectrum beta-lactams in treatment of Pseudomonas aeruginosa infections, can also be justifiably included on the restricted list. Items omitted from the restricted or unrestricted list would generally not be stocked, although they can be obtained on short notice if necessary. Such a policy should have a mechanism where desirable new agents are added as they become available and is most appropriately decided at a therapeutics committee. Policing such a policy is best effected as a joint arrangement between senior pharmacists and microbiologists. So again, this is a scenario like where we had in the institution where I worked. We had two lists, one restricted, one unrestricted. And on the restricted list, we have several agents, and you could not prescribe them because first, the cost was very high, okay? And then we were not stocking them in large quantities. So for you to get them, we had to we needed to be sure that your patient was aware of the cost and they were actually able to pay. As you know, in Kenya, most people pay out of pocket. So this combined approach of both restricted reporting and restricted prescribing is extremely effective and provides a powerful educational tool for medical staff and students faced with learning the complexities of modern antibiotic prescribing. In some hospitals, antibiotic teams have emerged to advise and educate staff while monitoring compliance with prescribing policies, as well as ensuring good standards of patient management. So that's it for antibiotic policies. Next, I want us to look at disinfection policies. Uh, Disinfection describes a process that eliminates many or all pathogenic microorganisms except bacterial spores on inanimate objects. Disinfectants are antimicrobials applied only to inanimate objects. This is different from antiseptics, which are used only on the skin and not for surface disinfection. Disinfectants are not used for skin antisepsis because they can injure the skin and other tissues. So it's important for you to be able to separate the two. What are disinfectants? What are antiseptics? Okay. And as we've said, disinfections, inanimate objects or surfaces. Antiseptic, they are used on the skin. The aim of a disinfection policy is to control the use of chemicals for disinfection and antiseptics and give guidelines on their use. Availability of many disinfectants available allow considerable scope for choice and inclusion of agents in a policy to be applied to such areas, such as industrial plants, walls, ceilings, floors, air, cleaning equipment, laundry, and to the extensive range of equipment in contact with hospital patients. So again, think of the newborn unit we were talking about, okay, and the spread of Klebsiella. How do you think it spread? Apart from probably being transferred by the clinicians and nurses who were handling one baby, finished, did not wash their hands or disinfect, moved to the next baby and transferred the infection. Okay. Again, how was cleaning being done? When you discharge one patient, okay, what do you do to prepare the bed and the room for the next patient? Okay, those are some of the things we are talking about. Disinfection, we are looking at laundry, okay, extensive range of equipment, maybe those the incubators, okay? How do you prepare the incubator for the next patient, okay? So This involves disinfection policies. The control of microorganisms is of prime importance in hospital and industrial environments. In the pharmaceutical industry, contamination of the product may lead to its deterioration and to infection in the user. In the hospital, there is additional consideration of patient care. Therefore, protection from nosocomial, which are hospital-acquired infection, and prevention of cross-infection must also be covered. Hospitals generally have a disinfection policy, although the degree of adherence to and implementation of the policy content can vary. So again, remember we talked about all these sensitive areas, okay? We're not just talking about the ward. We are talking about the NICU, we are talking about the ICU, we are talking about burns unit, we are talking about HDU, okay? All these areas, we need to know how do you handle them, okay? Or even theater, okay? How do we ensure that we use the right disinfectant so that we are able to kill all the microorganisms that may be present, okay? So the core infection control committee comprises of the pharmacist, the consultant medical microbiologist, and senior nurse responsible for infection control. This committee should formulate a suitable policy. So again, remember, as we've said, first you identify what are these key areas, okay, before you even think about the policy. What are the key areas? What are disinfectants are going to be used, and are they going to be used indefinitely, one disinfectant for the whole year, or what are you going to do, okay? All that is going to determine whether you're going to have a successful disinfection policy. So this core team may usefully be expanded to include, for example, a physician, a surgeon, nurse teachers, nurses from several clinic areas, sterile services, manager, domestic, superintendent, purchasing may also be represented. Okay. The expanded committee will meet regularly to help with the implementation of the policy and reassess its efficiency. So again, of course, as we are talking about, we identify these key areas. You're not just going to talk about the newborn unit, and you're not involving the people who are working in the newborn unit, okay? You need a representative there to be able to enforce what you are, the decisions you're making, to make sure that if we are saying that we are going to be rotating disinfectants, how are we going to do that, okay? The committee may give a choice of disinfectants in many of the areas covered by a policy. However, in certain instances, specific recommendations are made as to the type, the concentration, and the usage of disinfectant. Uh, Categories of risk to patients may be assigned to equipment coming into contact with a patient. This risk dictates the level of decontamination required and degree of concern. So articles are divided into three categories based on risk. We have high risk items, intermediate risk items, and low risk items. Of course, you're able to appreciate that you make this division so that you can actually decide. There are some items that are going to come direct into contact with the patient, and when they come into direct contact, you can appreciate some of them will come into direct contact with broken skin, others will come into direct contact with unbroken skin, others will not come into direct contact with the patient, okay? So it's important for you to be able to appreciate this, okay? So that if you're in, say, in part of that committee, then your risk, intermediate and low risk. We have high risk items. These have close contact with broken skin or mucous membranes, or are those introduced into a sterile area of the body and should therefore be sterile. They include sterile instruments, gloves, catheters, syringes, needles. For these items, liquid chemical disinfectants should only be used if heat or other methods of sterilization are unsuitable. Then we have intermediate risk items. These are in close contact with skin or mucous membranes, and disinfection will normally be applied. Endoscopes, examples include endoscopes, respiratory and aesthetic equipment, wash bowls, bedpans, similar items are included in this category. Then we have low risk items or areas. This include floors, walls, okay? They are not in close contact with the patient, but you can appreciate like for like walls and floors, okay? They need to be clean, because otherwise, if they are not, it's possible that you could have spread of infection. Cleaning here is important, with disinfection being required, for example, in the event of contaminated spillage. Okay, so again, it's important for you to be able to appreciate what kind of risk item are we talking about. Okay, any questions? Okay, if there are no questions, that brings us to the end of this unit. Apologies, today's class was very rushed, but it looks like getting makeup is becoming quite a challenge. So, yes, kindly go through all that we have done today, hopefully at a more relaxed pace, and it will all be able to make sense. But just it's important for you to appreciate, because if you end up in a hospital as one of the committee members, at least it would be good for you to appreciate what we are talking about. Okay, working in a hospital, you should be able to differentiate the different areas. What are the risky areas? Why are we concerned about the newborn unit, the burns unit, the ICU, okay, high dependency unit, okay? Because these are patients who rely fully on the uh, clinical staff, okay? And if the staff are not careful, it's possible for them to transfer infection from one patient to the other, okay? And so that's why these policies are actually very important. Remember what we have seen about the KNH issue with the newborns dying in the newborn unit? Quite unfortunate, and yet it's a matter of infection. Okay, so if there are no questions, we'll stop there for today. I got communication from your class rep that we will have our cut on 23rd and 28th. Okay, so kindly prepare for that. Okay, sawa sawa. So have a good afternoon and all the best.